Yes. Also, sometimes we do get mixed infection, because you can see both chlorotic and mid-rib yellowing. We can report we have already reported grassy shoot disease as well as this white leaf disease together in the clump, and there we reported more infection. In the second picture, you can see that is because of mixed infection of sugarcane yellow leaf virus, sugarcane yellow leaf phytoplasma, and ratoon stuntic disease. You can see even after 6 months of stage, there is no tailoring, and there's a maximum loss is there. So these are the very important diseases. Next, sandal spike already so I need not take, because next our my colleague Dr. Vikas Mandal will discuss more in detail. It is again a very important old disease, and we have been no solution, no 100% reported vector for this disease, and whatever vectors reported, they are not confirmed. But molecular identity is confirmed now. Earlier, only known on the basis of electron microscopy and insect transmission, we suspected plasma association. But now it is 100% confirmed with different multilocus gene and ribosomal gene. That 16SR first B subgroup is associated in India in all southern states, Mysore, Tamil Nadu, Andhra, wherever sandal is grown, we reported first B association. Next, please. Now bamboo is again coming in a big way. Now it is becoming a cash crop. Earlier we never considered bamboo as any significance, but we have all northeast region. There are a huge plantation, huge hectares of plantation of bamboo, and we have recorded from different states, different types of symptoms, especially witch's broom. That is from first report is from Pune by Amit Yadav. Is reported as second C group association, and recently we have been working from Tripura, UP, and other parts of India, and we reported second D association by utilizing 16-RNA gene and TUF gene. So it is again very important crop nowadays. Government is also promoting bamboo plantation, and phytoplasma is becoming a serious problem. So earlier, there is no other report from the world except from another bamboo species on Phyllocystis nigra from Korea. Next, please. Rice, already Dr. Biraktamat has said, that about they discuss nephrotetics that only breeds on rice, and that not only transmit rice drop phytoplasma, but rice tunguro virus also. In India, we have reported association of, there are two different phytoplasma diseases in rice, rice orange leaf and rice drop disease. In India, we have reported from Tamil Nadu, Coimbatore, association of 11th B subgroup, and from Bahraich, from Uttar Pradesh. And we reported association of pituitary vectors, we can say, from Riselia zigzag leaf hopper. Is again very common in Tamil Nadu. And one of our colleagues, they seabird, and reported Riselia dorsalis as a vector. And we reported nephrotetics from UP, Bahraich area. That is a major rice growing area in Uttar Pradesh and Nepal. Next, please. So you see all the important, only I'm highlighting the most important cultivators species. Earlier, there was only 2010, there was a report from China of phytoplasma known as wheat blue dross disease. Since then, no report was there. But only 2011, we got information.
Dr. Rao, can you just wrap it up in a few minutes?
Oh, okay, sir, sure. These are wheat, again, we have recorded association of 11th and 14th group. We also recorded pituitary vectors from central region, that is Madhya Pradesh, our regional station at Indore. We have recorded from 170 accessions of durum and bread wheat from Indore. Next, please. We also reported several vectors, but still is to be proved to be a natural vector. Next, please. Maize leaf. We have reported maize leaf dross from Jammu area. This is again reported to be associated with first of phytoplasma. Next, please. Please proceed the slides little fast. Season phyllodes, that is the most important. If you can say this is among the top 5 diseases in India, and you can We surveyed all the sesame growing regions of India and report association of three. First B subgroup, second C, and second D. Second D is the most dominant spread species of Phytoplasma among all the sesame germplasm. There is no resistance source available. So still for the last 2-3 years, we started working on vector, and we have reported Orosius and Empoesca and Maresca as a very effective insect sources. We also tried to maintain culture of sesame through grafting in this periwinkle. Next please. A new method of grafting, leaf disc grafting for establishment of culture. Groundnut we have reported from Andhra, again very important. It is this for the last 2-3 years we have been recorded very severe incidents, and we have recorded association second C subgroup of Phytoplasma. Next please. Soybean also we recorded from Delhi. We also reported alternative host and some putative vectors. Next. Thoria we have reported. Here you can see we have reported a plant hopper. Lateral Strialis as a important role in transmission of Thoria phyllodes. Next please. Vegetable, you can see a number of vegetable species we have reported, more than a dozen of reported to be affected with first, second, and sixth group of Phytoplasma. You can find brinjal, tomato, potato, lactuca, cabbage, and cucumber. They are host of different groups of Phytoplasma. We reported from India. Next please. So you can see the diversity of Phytoplasma in vegetables we reported from Orissa. Next please. Next please. Tomato big bad, as I told you in the beginning, it is become really, you can see earlier we 2015 we first of all reported from Telangana. Now you can see it is spread all over northern and southern part of India. So it is really spreading very fast. Next please. We have to work on this vector and other means of transmission. Brinjal, already we discussed a lot. Second D and sixth group are the most dominant species of Phytoplasma. We reported Histimoss as a natural vector transmitting this Phytoplasma in most of brinjal plantation in India. Cowpea beeches broom, you can see different types of symptoms, 100% loss, and this is, you can see different types of groups and subgroups associated. And we confirm association of Phytoplasma by applying ribosomal gene and other multilocus genes in different parts of India. Next. Legume species, you can find pigeon pea, cowpea, lentil, chickpea, all they are susceptible to Phytoplasma with different. We got association of 9C group, 6D, and 14A group. Chickpea is again coming a big way emerging problem all over India, and we found association of Phytoplasma, Master virus, and cucumber mosaic virus. It is again very serious. We also identified some leafhopper species, Ambresca, Empoesca, which are important in transmitting this disease, Phytoplasma, from healthy infected to healthy chickpea plantation. Next. We also develop multiple system. In one PCR cycle, we can identify, of virus and Phytoplasma together. We developed a protocol of multiplex. Instead of going for first round, nested round, and
Dr. Rao.
Next, please. Please go.
Now, please pick up.
Yeah, please. Next, please. Next.
थोड़ा time ज़्यादा हो रहा है.
Sir, just I'm thinking, already is.
Okay.
We also identified in different ornamental. It will discuss further by other speakers. So he will cover ornamental species. Next, please. You can see the marigold again, very serious for all over India now. All the marigold growers, they are facing this problem, and now we have to work on which particular insects are responsible in different states of India for marigold Phytoplasmas. Next, please. Chrysanthemum, already in Delhi we have reported different. You can see phyllodes, no flower, no commercial value. They completely lost their market value because of Phytoplasma association. Next, please. Rose cultivars, again we have reported. Next, please. Yeah, we also developed coconut. Already we discussed. Erica nut. Next, please. And oil palm. They are again very important host of Phytoplasma of 11B subgroup. So it is again very spreading all over South India. And still we don't have any vector species known which are really playing a role for transmission of these phytoplasma. Next, please. Next, please. Already next. Fruits, no, you can see a variety of fruit species, mango malformation. Next, please. All we have reported different species. Papaya, you can see shoot proliferation, pelodi in papaya from Kerala, Faizabad, and other states of India. Next, please. Pineapple, Tripura. Next. Japota is again very important in all over Karnataka, Kerala, and Tripura. We found flat stem and pelodi symptoms. That is minimizing the in all old orchards. We are in citrus decline is also we reported association of phytoplasma, citrus greening, and citrus dystegia virus. Next, please. Banana, you can see banana earlier only banana bunchy top virus was there, but we reported association of phytoplasma. And Mysore banana is Mysore virus with the banana bunchy top and colossus symptoms in Tripura. Next, please. That is Gigifus enophalia from Karnataka. We reported a fifth group association. That is the new group from India. Next, from cashew, we reported from Tripura. That is the cardamom from Kerala. Next, that is elephant foot yam from Tripura. Next, please. And from other places, Uttarakhand and Uttar Pradesh. Some tree species also like sandal. Some of the forest and natural urban trees are also reported to be host of phytoplasma. So we reported Acacia magnum from Tripura as a host of first group, second C subgroup, and other tree species like Serrella, tuna, neem, and other this spum species has been recorded as a host of phytoplasma from different states of India. Next. Seed transmission already spoken, very little is known, but in India we have two incidents recorded: Celosia ornamental plant and embryo transmission of coconut root tubet in India. Next. This is the vector, that is, I would like to take 1 minute, as Viraktamat said, now we have little better information available for some true vector species, like Chrysanthemum, you can see some are putative vectors, some of the, but we have to confirm. Most of them, they are putative vectors, but we have to confirm through transmission assays. But now we have a more list for putative vectors and natural vectors in India. Next, please. Weeds are again playing very important role for transmission through the help of insect vectors. Next, please. These 6 species are really important. All over India, these are the major weed species which are the natural host reservoirs, Parthenium, Kilium, Cannabis, Datura, and Phyllanthus. These are the real, and you can see association of different groups of phytoplasma. So they are reported to transmit different groups of phytoplasma from wheat to cultivated crops and vice versa. Next, please. Mixed infection also we have reported number of cases, for different viruses, different groups of phytoplasma and bacteria. Next, please. This already very little is known host metabolic interaction. Previous speakers are already there is no machinery, phytoplasma. They have to depend on host metabolism, for their survival and multiplication. So very little attempt has been made in India. So that is again a major challenge before us to work on. Next, please. Management approaches, still we don't have, we have different types of management approaches, but nothing is 100%. We can awareness is very important. Now we are trying to develop some resistant varieties. Vector control, as suggested by Dr. Viraktamat, is very, very important. Unless until we can't manage plant to control. Some antibiotic treatments are there. Tissue culture, apical meristem culture we are applying, and some of the hot therapy and quarantine regulations. Next, please go through very fast. We have developed some in vitro and in vivo method for application of tetracycline for management of Chrysanthemum. Next, please, and marigold phytoplasma. And we got by applying a T per liter of oxytetracycline, a very good control of phytoplasma remission in the different varieties of Chrysanthemum and marigold. Next, please. Next. So now we have to work further, no? But again, there is as Dr. Krishna Kumar suggested, there is a limitation. How many times we can apply, how much amount of oxytetracycline we should apply? No, it is not possible. We have to have some alternative approach. Next, please. For controlling, we also developed some in vitro method for meristem tree culture for very good control of Phytoplasma in ornamentals, especially in ornamentals. Next, please.
Kindly wrap it up in a minute.
Two slides more. Next, please. Already I have discussed, no? So both we are applying in vitro and in vivo method by applying oxytetracycline. And for insect vector, we have also suggested to use tetracycline plus insecticide. And next, please. In marigold, we planned an experiment and we got very good control. Next, please. By applying tetracycline and this imidacloprid combination for management of Phytoplasma under condition. Next, please. Next, no. This already this is a positive results. We can plan further on the basis. This is just a lead, no. This we can plan further to control different strains of Phytoplasma in different crops. Next, please. So now that I will take one very important, sir. This quarantine, still we have 172 plant species, but still some of the Phytoplasma strain from America and Western countries and Middle East we don't have. So it must be treated as a very right time to have awareness to restrict the entrance of these pathogens. No, for line which is grown from Middle East, palm lethal yellows. Krishna Kumar has already initiated in his is very important in Caribbean and America, South American countries. Coconut yellowing, no grape vine yellows, as suggested by Dr. Biraktamat. All over Europe and America, Australia is a very serious apple proliferation or European stone fruit Phytoplasma. Again, is of great quarantine concern. No, it should be taken. Wheat blue drop disease from China, papaya bunchy top, sugarcane yellows. There are other strains only reported in Mauritius. In sugarcane is not in India. So these are some of the future aspects we must consider: culturing, epidemiological studies, seed transmission. And we have a very good active cooperation coordination in India. More than 15 centers are involved. We are coordinating and collaborating for progress of Phytoplasma identification, vector, and other aspects. We have recently published covering all aspects of development of Phytoplasma worldwide, and we are also publishing a journal. Thank you so much. I'm sorry for a little delay, and I must thank to my working team, my colleagues working in India and abroad for their collaboration and contribution. Thank you so much.